Hello, everyone, and welcome to the magical seventh episode of Honest Discussions. I'm your host, Dr. Randon Patterson, and this episode is magical because I get to talk to a man after my own heart, Dr. Kas Thomas, who is a citizen researcher. Kaz is doing some pretty remarkable work on moonlighting genes and how many of them may be encoded as antisense within organism genomes. Uh, that is heresy to a number of molecular biologists, but I encourage you to listen in. Some pretty reasonable explanations for why this might be happening. We also discuss what it's like to be a citizen researcher and how not having the constraints of industry or academia hanging over your head can actually lead to whole new areas of research. And this has really only become possible in the last 10 or 15 years because of the availability of genomic and proteomic data sets online. It is the beginning of the rebirth of the citizen scientist. And that's something that this platform really promotes. But before we get to the show, I'd just like to encourage everyone once again to please like, share, subscribe, help us get to that 1,000 subscriber mark. We are growing and every little bit helps. And I promise once the channel is monetized, it's going to be better than ever. So on that note, on with the show.
Wow, there's so much here that you're hitting on so many of my favorite topics. I barely know where to begin. Yeah, so I look at myself as kind of an outsider citizen scientist. I do have academic training from University of California at Davis from many years ago.
I was there for a few years
myself. I spent a career in technical. Well, there you go. That's a great place. Fantastic.
It is, actually. It is. I love
Davis. Education. It's a great campus, and it's a great institution. And I'm still in touch with my professor. I left graduate school 48 years ago. I'm still in touch with my professor. John Ingram is 97 years old, and he still answers my emails. So anyway, I left science, spent a career doing technical writing in enterprise software and desktop publishing for a while back when that was a thing. But now I've returned to science as an amateur bioinformatician. And I find that coming at it with kind of an outsider viewpoint coming back into the field like this gives me a huge advantage. And yes, to return to your topic, I mean, the paper does deal a lot with antisense, and I would love to get into that more. But to answer your question, I do a lot of messing around with bioinformatics, and I'm just curious to learn how genomes work, how genes work, how codons work, how regulation works. I view myself as a person who uses comparative genomics to get insight into integrative physiology types of questions. I want to understand cell biology. I want to understand the big picture. So I do a lot of top-down type of work and ask systems level questions. But I do a lot of messing around.
Language. You are speaking my language. I mean, we have a similar background. I mean, I retired at 43. I retired really young. And now that I have been away from academic science for a decade, in the last 10 years, I have done my own research. Now, personal, completely, never with the intention of publishing anything, because it was only for me to learn. And as I left academia, I was hosting systems biology conferences, and I mean, I'm also a top-down person, but fully aware that without the fundamentals being correct, you can't build a pyramid. So
That's right.
yes, understanding the intricacies of just the smallest pieces is still required to see the big picture.
Absolutely.
And
But it's you got to come from both directions, though, because otherwise you'll never get there. If you just focus on the minutia and the esoteric edge cases, it's hard to synthesize a complete picture from that. So you have to ask the top level questions as well as doing the bottom level work. Otherwise, you never get there. And you have to keep an open mind the whole time. And you have to be curious. I mean, that's what makes a good scientist, is just curiosity. And you have to be fearless about asking dumb questions. You have to take the attitude there is no such thing as a dumb question. And you have to ask lots and lots of dumb questions and just, engage with your inner child to bring out these simple questions in order to get really top level knowledge. And I do a lot of experimenting in the computer just with my own code. I do a lot of poking around in genomes. I do a lot of quick experiments just to educate myself. And I was fooling around, to get back to your question, I mean, I was fooling around one day with just some basic codon statistics, looking at tuberculosis, so one of my favorite organisms, Mycobacterium tuberculosis. I was actually doing a lot of looking at palindromes in DNA, which are everywhere, and the kind of like the elephant in the room. Nobody wants to talk about all of the palindromes that are in protein coding genes, within the genes, not in the control regions, but within the genes. And I was looking at that, and I was doing some simple enrichment experiments to see, well, what kinds of genes have palindromes? And as I was eyeballing the results and looking at the annotations on the genes, I kept seeing what I thought were a lot of familiar genes that I would say to myself, now, wait a minute, isn't this a so-called moonlighting protein? And, I go down and I see, glyceraldehyde 3-phosphate dehydrogenase. I would go down and see enolase. I would see DNA K and various chaperones. And I started seeing so many of these. It suddenly came to me, these are moonlighting proteins.
Well, maybe we should talk a little bit about what a moonlighting protein is and what a palindrome is.
Right. Well,
so palindrome is just the sequence reads the same in the forward direction as the reverse direction. But
in English, that's how it works. But of course, in DNA, you've got a complementary strand. And so in DNA, a palindrome is a sequence for which the reverse complement is the same as the original sequence. And so GGCC, the reverse complement is GGCC. And right. that makes it a palindrome in DNA, in the DNA world. And the significance of palindromes is that they can form alternative secondary structures. They can form so-called cruciforms and hairpins and cloverleafs and all kinds of secondary structures. So a typical example of that is a transfer RNA has a bunch of palindromes in it, and that's what gives it the kind of cloverleaf looking structure.
Yes.
And the orthodox belief in the world of genomics, there's a lot of orthodoxy in science, and one of the orthodoxies that bothers me is there's this orthodox belief that all DNA is always B-form, canonical, Watson-Crick DNA. And of course, we're finding more and more exceptions to that. But people say, well, because of that, and because it takes you have to cross an energy hurdle in order to get a palindrome to form a cruciform structure. These secondary structures don't just spontaneously form in vivo. So people say, well, these don't exist in vivo because it takes energy to form them. Once they're formed, they're stable. But these probably don't exist in vivo, which is nuts. It's completely incorrect. I'm just saying there are ways to melt the DNA in vivo that happen all the time because RNA polymerase creates regions of positive supercoiling ahead of it on the DNA, and regions of negative supercoiling behind it on the DNA. And you can well imagine if you have divergent transcription, you've got two regions of negative supercoiling going on. That's going to unfold the DNA. There are all kinds of ways for DNA to locally melt and form secondary structures in vivo. And this is a ripe area of science. And I was looking at that when I got sidetracked by moonlighting genes. But to answer your other question, a moonlighting gene is just what it sounds like. It's a moonlighting protein is a protein with more than one job. So moonlighting proteins violate the orthodox belief that one gene encodes one protein with one function. So one gene, one protein, one function is kind of the baseline orthodoxy. It's completely wrong, of course.
I would hope that orthodoxy is dead. I mean, that's one.
I would hope so. And yet we have this whole phenomenon of moonlighting genes, which we still treat as some kind of exception. That's some kind of weird thing that's the exception rather than the rule. Okay, but you know, the very term moonlighting genes, moonlighting protein is a protein with more than one job. Well, don't most proteins have more than one job?
I would argue that every protein has more than one job.
Yeah, I mean, pleiotropy is the rule, not the exception, right? I mean, and you know, in higher organisms, lower organisms, genes can produce. Well, in higher organisms, they can produce more than one kind of protein because of how the introns and exons can recombine. But even once you produce a protein, it can easily have more than one job, right?
Yeah, I mean, that's my point is I think every protein has multiple jobs, and that if it's truly a protein network, then the hub proteins within the network are going to have multiple jobs. And one of the proteins you've already brought up, GAPDH, it appears everywhere. It appears to interact with everything. So much so that people want to say, oh, it's just a sticky protein. And that the reason you see it in your yeast 2 hybrid experiments, or the reason that you see it in your immunoprecipitation experiments or any type of pull down of protein complexes, is because it's just a sticky protein. I would argue that the cell is much smarter than that, and that proteins like GAPDH, which make NADH, which is a form of energy and a signaling molecule, all rolled into one, and potentially even a scaffolding molecule, that of course GAPDH would have multiple places that it impacts, because not only does it itself serve as a scaffold. I mean, we can just talk about GAPDH. How many functions does it have? Well, it has been shown to work as a scaffold.
And it has been shown in higher organisms to play a role in apoptosis. If too much GAPDH accumulates on the outside of the cell, it's a suicide signal.
It's also the same thing. I actually published a paper on the same thing with the IP3 receptor. So while GAPDH is forming on the outside of the cell, at the same time, it is forming on the inside of the cell. It's binding to the IP3 receptor and increasing or decreasing its sensitivity to calcium so that it won't shut off when it should, which is also part of this moving to the apoptotic signal. So I didn't know it formed on the outside of the cell till just now, but given what I know about the inside of the cell, makes perfect sense.
Yes. It's amazing how many of these gene products have roles on the outside of the cell. And we are still fascinated by this, and it's kind of anomalous, not well understood. And so there's this whole database of moonlighting proteins, moonprot.org, I think it is, and there's hundreds of examples of it now. And so we just got fascinated by that. We basically accidentally discovered that we could create an enrichment, a bioinformatics enrichment, that would enrich for moonlighting proteins. That struck me as like, wow, I didn't even know that was possible. And I thought, so how can I refine that? How can I make other enrichment algorithms to enrich for those so that I can understand them better? And that's how we kind of got started down that road. But it was a long, fascinating road. Took us into antisense, took us in various directions, and now we're just overloaded with low-hanging fruit right now. This is a great time to be doing bioinformatics because there's so much orthodoxy, and people are not looking at things they should be looking at. And so it opens the door to people like you and me to come along and say, well, I'm going to look at that if you're not going to look at it, and there's just so much to do. It's just unbelievable.
There is. I mean, part of the reason that I retired early is I was being denied the ability to go down some of these rabbit holes that you're going down. It's when you have dogma within any field, and bioinformatics is certainly not immune from it either.
Right.
You can't move forward because you can't get the work published. You can't get people to even believe what you're doing. And now I had the benefit of being a cell biologist and a biochemist. So, I mean, at least I could validate some of what I was doing. But I mean, my predictions in the mid 2000s that were published were that every protein was multifunctional, that there were bioinformatic solutions to finding these functional domains and predicting their function, and was going along really well until we get to a point where you run into the brick wall of, well, we can identify these 70%, but these other 30% clearly were missing information. There's something that we're not taking into account, and therefore we need to start pulling from other information sources. So when I proposed this, that basically what we could obtain from sequence data alone had been exhausted, and that we need to increase the amount of data once again of what was going into the measurements. Well, that's not okay, because the dogma is that it's all in the sequence, and if you can't do it just from the sequence, then you don't know what you're looking at. And I was like, no, there's so much more within those sequences that we can add data. We can add their physical attributes. We can add things like you're doing right now, where you see repetitious patterns, or you see an enrichment for certain motifs. And now that was somehow that goes outside of the dogma, because you have to be able to pull it all from the sequence. And my argument is you can keep adding more and more information into those sequences, and therefore
yes,
you will get a better measurement the next time around. Completely stonewall.
When we talk about those sequences, I mean, people are still talking about the main sequence of the gene on the message strand. They're completely ignoring the reverse complement strand. The current orthodoxy is that, there's not enough bandwidth in DNA to encode information in both directions. That's absurd. So there's still this kind of belief. Yeah, there's this kind of belief still today that from 50 years ago, that antisense equates to nonsense, or that antisense isn't transcribed. If it's transcribed, it's just a non-coding RNA. But certainly they're not translated, you know, and there's all these, oh my goodness, there's all these orthodox beliefs that are holding back science, because this is extra data and extra information that needs to be considered. There are thousands of antisense open reading frames in almost every genome, and these have to now be seriously considered, right? And we got into an argument with one of the reviewers on our paper who just could not abide the idea that we were suggesting that these antisense sequences were actually translated. I mean, he admonished us and said, you know, there may be thousands of these things in the genome, but very few of them are translated. And I challenged him. I said, on what basis, on what data do you say very few of them are translated? I mean, shouldn't we just take the very simple point of view that an open reading frame is an open reading frame? An open reading frame is a sequence of DNA that is translatable in theory because it meets the criteria for translatability. That's not to say that it is translated or is not translated, but it meets the criteria.
The same is
true. And my personal view is
your personal idea.
Yeah, I mean, and the cell doesn't matter. Yeah, well, my view is that these decisions of to translate or not to translate are done by inanimate objects through physics, through physical and chemical interactions. It's not guided by anything. It's all physics. I mean, the cell does not know the difference between one open reading frame and another. How could it? How could it? I mean,
see, this is where let me push back just a little bit there, because it can know because the machines are designed to know. So the
machines are designed. I think a properly nuanced rephrasing of what I just said is that all open reading frames are potentially translatable, and I like to look at it that they are all translated. It's just a question of efficiency. Some are translated with such low efficiency that you might as well say that they're not translated. Others are translated with very high efficiency. And my point is that they're all on a continuum. It's all on a continuum. And some of these will be translated more efficiently than others. That's all. That's all we need to say. But these processes are all inanimate objects. I mean, you know, a ribosome is not sentient. It doesn't know what's a good open reading frame, what's a bad open reading frame. It will either stick to the messenger and complete the process, or it won't. It will be highly processive, or it won't be highly processive.
Well, if you ask some physicists, they will say that your ribosome is conscious because the entire universe is built on consciousness. However, I agree with you that there's a continuum of translation, that there are some proteins that maybe only get transcribed 5 or 6 times, but those 6 proteins might be completely sufficient to deal with whatever or the reason that they're actually being translated. This is a question I really want your answer to, is what % of proteins do you think have been discovered of all that's out there?
Well, you're getting into one of my favorite topics, because our very next paper that I'm writing up right now has to do with the dark matter of genomics, which is to say hypothetical protein genes. Hypothetical protein genes are numerous, and in higher organisms they can be well over half the genome. And hypothetical protein genes are everywhere, and they've been a problem for genomics since the dawn of the genomics era. And we still don't know. I mean, look at tuberculosis, a very well studied pathogen.
Perhaps 25%
of the, that's right, and 25% of the genome consists of hypothetical protein genes about which we know very little. That's unacceptable. We can't have 25% of the genome of a major pathogen be completely unknown. What is that? That's madness. You know, we've got to address that. So we're doing a lot of work right now on hypothetical protein genes, taking into account a lot of the ideas that you're talking about the sequences and looking at, of course, looking on the reverse complement strands. And we haven't figured out all of the, we haven't even gotten started figuring out the functionality of these hypothetical protein genes, although we have some ideas based on co-enrichment with other proteins. But we do think we've figured out where they come from. We do think we know the dynamics of how they form and why they form and why they tend to be a variety of different lengths, although they tend to be shorter than other proteins.
Just off the top of your head, how many proteins do you think we've really, or what are the percentage of proteins that you think we found versus what's really out there? Just a ballpark.
It's hard to know, but I get the feeling that it's a tiny fraction. I mean, we've characterized maybe 1 to 10%. 10% is optimistic, I think. Maybe 1% of what's out there.
And that's also what I think. And that is something that I think a lot of people would argue with us over, like that
yes, absolutely.
This is the problem I have with some of the established fields like this. Like, this is a very established field. We do know a lot, actually. It's amazing how much we do know about DNA and RNA and the processing of it and how it works, and new functions for RNA that we have found. I mean, we do know a surprising amount. However, just because we know a surprising amount doesn't mean there isn't some incredibly surprising amount over and above what we know that still exists to be found. And this is one of these areas where I can't say it enough. 20 years ago, as I was sitting in Saul Snyder's office telling him I think that the IP3 receptor could have upwards of hundreds of unique binding partners, that he was just like, that doesn't make any sense. Like, we don't know of any proteins like this, that, and, kind of stay in the lane that we can publish in. And, you get out on your own and you want to go down that road, which I did, you can. But this is the reality of it. I think that by thinking that we know so much, we actually inhibit ourselves from getting to the next, what really is low-hanging fruit.
Yeah, I agree. I mean, one of my favorite idioms, and I'm being sarcastic, is gaps in our knowledge. We have gaps in our knowledge. Actually, we have knowledge in our gaps. The world is mostly gaps of knowledge. We need, like you said, what we need to know and what we're going to know is huge compared to what we do know now. And it's true, it's impressive. What we do know now is very impressive stuff.
It is.
But you have to keep it in context. We are, like you said, we are at the beginning of something here.
Exactly.
Right? And we have, yeah.
And what I
exactly.
think
we have a long way
your work is showing that it is, and what I think is really interesting from your study, on top of that you were able to develop this method for enriching these antisense open reading frames, which we're going to come back to, because I do want to discuss how you were able to find anything that resembled like a start sequence, like a shine sequence. And I do want to discuss that. But the proteins that you find that are encoded are shockingly similar. And maybe you could give me a little idea about why that might be and what might be going on with the fact that many of these proteins appear to have transmembrane domains, and if they're not transmembrane domains, they're lipid binding domains that allow for tight association with membranes.
That seems to be quite common, yes. Well, there's a very good fundamental reason for that, and this takes some, you have to almost draw it out on a piece of paper. Even I have to have trouble visualizing it, but very, very important concept here that I want to get across is that there seem to be open reading frames on the reverse complement strand of many, many genes, but it's important to understand that those reading frames can be in any offset from the start. And so they can be mod 3 equals 0, which means you just start looking at anticodons. You just start looking at codons from the very start of the 5' end, three bases, three bases, three bases. But they might start at an offset of 1, and then you start parsing 3 bases, 3 bases, 3 bases. Or they might start at an offset of 2, and then you start parsing. And so what we have found is that there are 3 different frames in which the open reading frame can begin. Now, naturally, once you're in an open reading frame, it's just all codons. But the point is that that entire open reading frame can be shifted slightly, either +0, +1, or +2, relative to the end of the so-called gene. One of the key findings is that these open reading frames, these frames in which the open reading frames exist, are non-randomly distributed, and the +1 frame seems to be preferred over the other 2, and the +2 frame is hardly ever used at all in any organism's antisense. We can return to that later, but to get more to the point of what you were asking, you have to imagine in your mind a codon on the sense strand. So the annotated functional gene has codons. For each of those codons, you have to ask what's on the other strand going the other way. And if the codons on the other strand going the other way, I'm going to go ahead and call them codons because some of these are open reading frames. They can be lined up such that base 1 of the top strand codon is over base 3 of the bottom strand codon, in which case base 2 is over base 2 on each. Or if they're slightly offset, if they're offset by +1, then what you end up with is base 3 of the top strand codon is over base 3 of the bottom strand codon. And bases 1 and 2 are over bases 2 and 1. When you have that configuration, it is a configuration that favors membrane proteins, basically, and nonpolar amino acids. So this all gets back to some very, very fundamental information that I wish was taught better in schools. Which there's 2 facts about codons that everybody needs to keep in mind always. One is
inform the public right now. What is it?
Okay, everybody listening, whether you're a student, whether you're a practitioner, whether you're experienced or not, keep in mind that base 1 of a codon tends to be a purine 60 to 70% of the time. This rule is called purine bias. All of the gene calling programs, Protocol and all the others, use this rule. I mean, they look for purine bias in base 1 of codons. This is a universal signature in bioinformatics across all domains of life. There are very, very few exceptions to this. I challenge anybody watching this to go find your favorite genome and start looking at all the genes and look at all the codons, and you will be amazed that they all, in the aggregate, you start looking at base frequencies, the first base of a codon tends to be a purine. It tends to be a purine. That's one fact that has to be kept in mind throughout everything. The And I think that's a basic cadence mechanism for the ribosome. I think that's how the ribosome stays in frame all the time, because it sees this boom, 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 this cadence of purine something, something, purine something, something. That's how it keeps its framing all the time. And it's apparently a very important mechanism because it occurs across all domains of life. The second fact that is absolutely critical to keep in mind is if you study the genetic code, it is very, very immediately obvious that when base 2 of a codon is a pyrimidine, the codon will encode a non-polar amino acid 80% of the time. So it's very, very important to keep that in mind. When you find a protein gene and you start analyzing its codons and you find that 60% of the codons have a pyrimidine in base 2, boom, you kind of already know that's a membrane protein. It almost has to be, because you know, a cytosolic protein is going to be water friendly. It's going to have a lot of you know,
charged and polar amino acids. Yeah.
it's going to have polar amino acids on the exterior. But the membrane proteins have to have a lot of non-polar amino acids on their exterior. So keeping those two facts in mind, you have to reconcile those two facts when you're looking at codons on one strand and codons going the other way on the other strand of DNA, and keep bearing in mind how they overlap. Are they slightly offset by +1 or +2, or are they just square with each other? Most of the time they're offset by +1, which means that base 3 of one will be opposite base 3 of the other. Base 1 of one will be opposite base 2 of the other. And the only way you can reconcile a 2/1 and a 1/2 situation is if the first base is a purine and the second base has a pyrimidine. And that means that genes that encode membrane proteins and membrane-friendly proteins, sticky proteins, if they have antisense open reading frames, those two will also tend to encode membrane-friendly proteins. And this is something that jumped out at us when we started looking at antisense open reading frames. We were like, wow, there's an awful lot of these that are at an offset of +1 from the other strand's gene. And why is that? And then we looked at, well, that's a very propitious thing for the alignment of these codons, given that base 1 has to be a purine most of the time. It all started to make sense. So we see that antisense open reading frames quite often are membrane-friendly protein products.
Well, it makes sense from the standpoint of if you're translating a membrane protein, you can pack more membrane protein information into that same sequence. It's already targeted to the places it needs to go to be around membranes to be translated. So from a energy perspective, it makes a lot of sense. Why would you make a whole new transcript that needs to go to the same spot when I can just send a multi-purpose one and it will get the job done? That brings me back to one thing you were talking about earlier. I wish I could remember it, but made me think of, the cell doesn't waste energy. It does not waste. So if you're seeing something being made or you're seeing something being transcribed, there is a purpose for it. It's not just making these things willy-nilly because, that's what you were talking about. You were talking about, well, the ribosome just translates what's in front of it. That's true. And how many it translates will depend on the stability. That's also true. But the cell is exceedingly smart in its innate intelligence of not wasting energy, which is its number one rule. And so
yes,
it's going to, you can envision where these, and even just within the moonlighting genes themselves, well, why would I make a whole new protein to do XYZ function when I could just modify one that already exists that has nearly the right chemistry already available to do this new function? And from an evolutionary standpoint, it makes sense. It makes sense that before you would go create a whole new protein, it would explore which proteins already exist that can do the job through modification.
Sure. Sure. Absolutely. And
so you had a model in your paper that I would love for you to just discuss off the top of your head about why these antisense proteins are getting made at the membrane and they're membrane associated, because this could have incredibly strong implications into potential for new antibiotics, potential for better understanding of transmission of disease-carrying molecules. I mean, there's a lot going on with that model.
Yes, and we think it's been overlooked for some reason, these various possibilities. But we are just seeing more and more handwriting on the wall that points to the idea that there are many, many antisense products that appear to be membrane friendly. It's a suspiciously high number, and they often occur, like these moonlighting genes, when you look at where they occur on the genome, I mean, they occur right next to genes involved in membrane and cell wall construction. And that just sent a red flag up for me. I was like, well, they're not randomly just sitting there. They are co-located with those for a reason. And we're seeing just a ton of handwriting on the wall that says, you know what, in growth phase, these genes, all of them, are probably co-transcriptionally translated. There's a lot going on in growth phase. Transcription, translation doesn't wait for transcription to be done. I mean, it's all going on simultaneously. And out of the ribosome is coming this protein product that before it even detaches from the ribosome, it starts doing whatever it needs to do. And if it's a membrane-friendly product and there's a membrane sitting right there, it's going to stick to it. And what does that mean? That means that there are potentially thousands of these things that are Velcroing the DNA to the cell membrane. I mean, there's a tether now of messenger RNA going down from the ribosome and the sticky protein down to RNA polymerase, down to the DNA. The DNA is tethered to the inner membrane all over the cell, at least in bacteria. And we think this may be a general mechanism, This has enormous implications, and one of the many, many implications of this is that if the DNA is tack welded, they're just kind of tethered and velcroed to the inside of the cell, well, now it becomes a gigantic mechanotransductive sensor of cell growth. And so as the cell grows and the cell elongates, it will pull open promoters and it will interact with secondary structures and pull open any cruciforms that are blocking the DNA polymerase. DNA polymerase has a sliding clamp. If it hits a cruciform, it's going to stop. It can't get past that.
Be knocked off.
Well, guess what? It sits there and waits until the cell grows a little more and pulls the DNA taut, and the cruciform goes away, and then the sliding clamp can go. So we think this is how the cell synchronizes DNA replication with cell growth, with physical cell growth. So yeah, we think there's plenty of implications here. It's mind-boggling, but yeah, we see all over the place, we see indications that the DNA is tethered to the inside of the cell.
please tell me an experimentalist is looking into this, because what you just said blew my mind, because I can just see it. I can just see it in my head, crystal clear. No, we've always known the DNA is associated in bacteria with the inner cell membrane. I mean, this is well established. It's part of why, when you're doing mini preps to get plasmid DNA out of bacteria, that you have to do certain things so that you don't pull down the genomic DNA, which is associated with the membranes. I mean, this is just something that every undergrad student is doing in their introduction to molecular biology. But given that it's known to attach, the mechanotransduction to me is just like, that just makes so much sense. Because now, as you move through the growth cycle, you get an unwinding of genes as they're needed. Because now I'm in the phase where I'm getting close to separating from my parent cell. So that requires a whole new set of genes to be expressed at a particular point. You don't need it on the other side of the cell. You need it here. And is the way the DNA is attached to the inner membrane, is that set? Is there a start point, or is there a point in that DNA where the daughter and the parent cell have the correct, the genome is placed in the correct area, so that when it's pulled apart, these genes are expressed, those create the cleavage furrow that you need, and so that the cells don't explode as they pull apart for the parent from the child cell. I mean, as an experimentalist, I can think of 20 experiments that you could do like this that would be simple and have an answer.
Yeah. Yeah, and I wish I could tell you that there are, hundreds of people working on this, but I'm not that well plugged into the community. I don't know if anybody's working on this, but this needs to be looked at very seriously. I mean, there are some palindromes in the mycobacteria. Tuberculosis has one PGRS gene that contains 59 palindromes of size 24 bases or longer, the largest of which is 88 bases long. So you can imagine a palindrome 88 bases long. If that thing ever formed in vitro, or in vivo rather,
you'd never pull
it would take a lot of pulling to pull it apart. Well, guess what? In tuberculosis, there's a well-known effect, and I've forgotten the name of the effect, but the effect was it was discovered that tuberculosis is actually an obligate aerobe. And the way this is discovered is you grow it in a flask, you put a stopper in the flask and don't let any more oxygen into the flask. Eventually, the culture will run out of oxygen and the cells will go into stationary phase. They will stop growing. And the amazing thing is, you can start them up again by reintroducing oxygen into the flask. But when they start up, they now divide synchronously. They divide synchronously, which means their DNA stopped replicating all at the same point. And we think we know exactly where that point is. There's that gene with 88 base long palindrome that's a replication blocker, and the DNA polymerase sliding clamp is just sitting there waiting for that thing to be pulled apart. So we think we can predict where to look for this. We know where to look for it. You know?
Previously, I was going to say, when you were talking about these palindromic sequences, well, the cell is also an expert at reducing the barrier of the energy barrier to do stuff, right? That's what enzymes are all about.
Right.
So to think that there's an enzyme out there that can lower that activation energy that's needed to, to break a palindromic sequence apart, that doesn't seem crazy to me. However, within the context of what you're talking about, it makes so much more sense that this is a mechanical, this is part of a mechanical signal transduction, not a classic secondary messenger type of signal transduction. And once again,
Totally agree with you.
if you were to take this into the mammalian nucleus, which is highly dynamic, has so many different membrane compartments that, you know, are little discussed because we don't know a lot about them, and has its own vesicles and has all kinds of stuff going on in that within the nuclear membrane, which then just naturally becomes a part of the endoplasmic reticulum where everything starts for transcription translation. It is there this there could be a mechanical signal within eukaryotic cells as well that and it just opens the doors to a whole new way of thinking. How many genes are controlled through mechano signal transduction in the nucleus? I would love to answer that question.
Yeah. Absolutely. Completely agree with you. And I would like to blow your mind one more time, which is...
Please do.
So on this same topic of mechanotransduction being the key to understanding how the cell synchronizes cell wall growth with DNA replication and probably a bunch of other stuff. You are no doubt familiar with the fact that Mycobacterium leprae, the cause of leprosy, is an uncultivatable bacterium that's never been grown in pure culture.
Unless you're in Florida.
To this, well, that's because we have a lot of armadillos. So if you go to a serious leprosy lab, which is a biosafety level 3 spacesuit type of place, but where the only way you can grow leprosy in a laboratory is in the footpads of armadillos. Did you know that?
No, I did not.
Yes, go look it up. Don't take my word for it. You can now use immune compromised mice also to put it in the footpads. It will grow and thrive in the footpads of immune compromised mice, as well as regular armadillos. We desperately need a way to grow that thing in vitro, right? And no one's ever succeeded. In 150 years, people have tried to grow it. It won't grow in the test tube. The speculation, of course, always is, well, it's got fastidious nutritional needs. We probably just don't know exactly how to meet the nutritional needs. That's not true. I'm going to tell you what I think is the answer. That genome is so broken. I mean, you probably know that the leprosy bacillus has hundreds of pseudogenes. It is a very broken, very genome reduced organism. It has only a fraction of the number of genes of tuberculosis that it's related to. It is missing 5 out of the 14 helicases that tuberculosis has. It is missing DNA repair enzymes. It is missing a lot of stuff. Okay, and I think, bottom line, the reason you cannot grow leprosy bacillus in a test tube is because it has lost all other ways of doing this. It needs physical manipulation to get the DNA to come open so that it can be replicated and transcribed and all the rest.
I mean, wouldn't that make it an obligate parasite? I mean, it's just a different type of obligate parasite.
Yes, exactly. And we think this mechanism is common in obligate parasites. But the reason you can't grow leprosy in the test tube is you have to massage the cell to get the DNA open in order to replicate it and transcribe it. And so if somebody will please invent an artificial footpad that does this, you could probably grow it in the lab, and that would be a historic event. The reason it settles into fingers, toes, foot pads, think about it. Those are high impact regions of the body. That's where a lot of mechanotransduction is going to happen.
You could build one of those for about $10 because
Yeah, that's
all
right.
you
That's
need
right.
is a little stepper motor that then
That's
you put
correct.
on a rod and put your pad of like a carbon nanofilm or something that has like really good properties that sells like it in general. And there's a bunch of that
Right.
stuff available these days, but I imagine you could probably still just use plastic, potentially.
Yeah.
Just put some curvature in it, but then just have it go up, keep going up and down at at
Oh,
different rates, and maybe you can get culture to form.
yeah, absolutely. I mean, if I had a starter culture, if I had a sample of leprosy bacillus, I would do that. Unfortunately, to work with leprosy bacillus, you need a biosafety level 3 lab with spacesuits and glove boxes and all the rest. So that's not
Yeah,
going to happen. So you would want to use something else, but you would want to take a very slow growing mycobacterium like Mycobacterium marinum, which is can be grown in the lab. It is safe to work with, but it's very slow growing, has a generation time of 10 hours or something. Yeah, if you put it in one of these type of apparatus, you will probably see the generation time shrink in half because you are helping the cell get through that difficult last phase of manipulating the DNA to the point where it can be fully replicated. So yeah, I wish somebody would build the apparatus. Like you say, it's probably 10 or 20 bucks. You go to get a fish tank water pump, and that's probably good enough. I mean,
right,
this stuff is
exactly.
not high tech.
If it grows on an
Right.
armadillo
So
foot pad, you definitely don't need something high tech. But
yeah, and
this this thought of the mechanotransduction, you could, and this is, now I'm just going out there, but how if there's mechanotransduction going on in eukaryotes, let's just say that there is, for regulation of DNA, how much would that be altered by changes in the membrane compositions of the cells? You could envision that getting the cells being too rigid could be completely detrimental, and at the same time the cells being too soft could be completely detrimental. And we know for a fact that, why do we have all these things, Lipitor and all of these drugs? Because they alter the lipid composition of our cells and have what it but do they really know how they're working? Not really. It's kind of like acetaminophen. We just take it that it works, and we think we know how it works, but in reality no one knows how it works. So could you see something like that? Could you see that a scenario where the changing of the lipid environment would completely mess up mechanotransduction through DNA?
oh, absolutely, absolutely. And I think the other another fascinating aspect of that is think how many viruses interact with the cell membrane, and think how much virus activity requires interaction either with the endoplasmic reticulum or some other membrane system. Now
It's
start to think about
universal.
what you're
It's
saying.
universal. No virus works without going through a membrane cycle. That doesn't exist.
Yeah, that's right. And so, yeah, when I start to think about that, I think, wow, what you're saying might have just tremendous consequences for dealing with viruses.
And then to take it back to the
Yeah. So yeah,
bacteria. Oh, keep going.
no. You go ahead. I know there's some
I was
delay
just
here,
going to
but
say, to take it back to the bacteria, if in fact these antisense proteins must be translated to enable a smooth growth cycle, then and they're still, I wouldn't say rare based on your results, but they're certainly much lower than what's coded in the positive direction, what we know or what's accepted as the direction. those could be excellent targets for disruption by small molecules as antibiotics.
Absolutely.
Because it may be hard and may be more difficult to evolve around than your standard single protein inhibitors. Because instead of inhibiting a single protein, if you're targeting this antisense region, you could be interrupting many proteins all at once, which is a much more difficult evolutionary hurdle for the bacteria to overcome than just creating a multi-drug antiporter and boom, everything's fixed.
Absolutely correct. We think that, for example, this general mechanism of, okay, you've got a primary gene going this way, but on the other strand, there seems to be these little genes for membrane-friendly proteins, some of which are going to stick to the membrane and kind of deliver the main gene into the periplasmic space. Well, you look at, heaven knows how many bacterial pathogens rely on periplasmic enzymes and transporters and antiporters, and a lot goes on in the periplasmic space of something like Bordetella or pathogenic Pseudomonas, or you name off, dozens of other organisms. And yeah, we think that this mechanism is involved in delivering all manner of periplasmic enzymes to that periplasmic space. It's the physical mechanism by which they just magically appear in the right place at the right time. And yeah, if you interrupt that with drugs or with anything, yeah, that could have a profound effect. And that could be, like you say, better than a broad spectrum antibiotic. This could be really a huge thing. Yeah.
Yeah. Like, that's why you said you're in Fort Lauderdale. Is that where you said you are?
In Tallahassee.
Or Tallahassee.
Tallahassee.
Okay. So I'm actually going to be coming through your area next month. And I'll be in Daytona Beach for like 2 weeks. I would love to sit down with you in person and have another round of discussions. I mean, we could, of course, talk about this, but I think there's so many topics because of your unique outsider perspective that we could get into. And I don't want to contaminate this particular discussion, because I know where a lot of that stuff's going to go, and it'll get pretty fiery. But as for this, so do you see this potentially having a role, or have you looked in double-stranded DNA viruses yet? Because they're a mystery, and they do interesting things, and I'm curious. I wonder if similar things are in these double-stranded DNA viruses that we just were never aware of, especially something like Mimi viruses. Or
yes, yeah, I too am fascinated with those. And short answer, yes, we're seeing some handwriting on the wall that says that antisense is a big source of viral new viral genes and viral genes in general. We're seeing that, as you know, DNA viruses have a very high percentage of hypothetical protein genes. And we think we know the reason for that. They I don't know if I want to divulge all the secrets, but that basically they arise by gene duplication. I mean, it's not rocket science. It's gene duplication followed by neofunctionalization. And sometimes when you duplicate a gene, there's an antisense open reading frame that survives, even though the duplicate doesn't survive. And that's very common, we find, because the antisense sequences are small, so they have a very small attack surface. The attack surface is all on the other strand, basically. So yeah, you would expect a duplicate the main gene will start to disappear. But if that antisense region is providing fitness to the cell or to the virus, then it may survive, and that becomes a hypothetical protein gene with unusual characteristics that we have not characterized yet in many cases. But yes, we think that DNA viruses are loaded with secrets that can be unraveled if you start looking at things in this particular way. Yes.
Well, now you have me wondering, obviously eukaryotic genomes, because I saw you kind of when I said, well, there's more forward than reverse, and you were kind of seemed like, well, maybe you don't agree with that. And I'll
No, I agree with that. Actually, we find that viable antisense open reading frames only occur probably on 20% ish, 20-30% of genes. So that
in bacteria.
Essentially what we're saying, in bacteria. So essentially we're finding what you're saying, which is that 80% of genes are just forward only, but 20% seem to have a very heavy involvement in antisense. Yes.
But if we were to look at a eukaryotic genome, if you were to just use the 80-20 rule on that, in humans, that would immediately mean there are 5,000 at minimum 5,000 new proteins that no one knows about. Minimum. Right?
Yeah.
So
yes.
Yeah. So have you looked in the eukaryotic sequences at all? Number one. And number two, given that eukaryotic cells are so much more complicated, wouldn't it stand to reason that in addition to transmembrane rich proteins, there would be whole other classes of proteins, potentially enzymes as well, encoded by these antisense ORFs?
Yes, we have only begun to look at eukaryotic cells. Paul is more of a eukaryotic person. I'm a bacteriologist, but in our recent work on hypothetical protein genes, we are looking at Saccharomyces. So we are looking at yeast. And yes, we are finding that the same mechanisms are used over and over across all domains of life. We're looking at yeast. We are looking at tuberculosis. We are looking at an uncultivatable archaeon, and we're looking at T4 virus, so a DNA virus. And we're trying to cross-validate and see how many of these ideas are at work across all of these. And yes, Saccharomyces has turned into an unbelievably rich experimental ground to look at these ideas. We're finding that even though it only has 10% hypothetical protein genes, which is low for a higher organism, that's really good. Still, we are able to see some of these effects that we're looking at, and it's clear that some of these effects are universal or pretty close to it.
Well, Saccharomyces still has a cell wall, so it's a nice eukaryotic organism to study because it's an animal cell. However, it has a cell wall. So for this DNA mechanotransduction idea, certainly yeast would be a great place. Plus, we know so much about it. We've one of the first genomes to be fully sequenced. One with it been in use by molecular biologists for, going on 50 years now. So we really great model organism to use. And the fact you're seeing it there, the only question then becomes, well, what happens when the animal cell transitions from having a cell wall to not having a cell wall? And then what does that look like?
Well, that's a very interesting question. We love those kinds of questions. You know, I've been looking at mycoplasma, which is kind of almost a bacterium without a cell wall. It is very osmotically vulnerable. It lives in the tissue space between mammalian cells and lives on the surface of the mammalian cell and so forth. And it's a very vulnerable little guy, and yet we find a lot of these antisense protein products that are membrane friendly. It's all pointing in the same direction. It's all saying that the DNA and maybe individual strands of messenger RNA are acting as sutures and acting as staples and acting as, you know, as stabilizers to stabilize the genome and stabilize the actual physical cell membrane so it doesn't explode. Yeah, we're finding a lot of this across a lot of different organisms. We think there's some fundamentals here that need to be looked at.
I mean, even within chromatin research and the way that DNA opens up to be read and folds, I mean, there's a lot going on in that field that they don't understand. And could it be that during chromatin remodeling, also during chromatin usage based on signals to start some important cellular cascade, there absolutely could be, as part of that mechanotransduction for giving the extra energy that they seem to be missing, like they don't understand at sometimes how an area of DNA is even able to open up to be read, yet it is. We know it is. We see the transcript. We see the protein product. Perhaps this is just in use all the time, every moment, and has been completely just unobserved until now. That is a real possibility.
I think you're absolutely right. Yes, because it goes to your statement that cells are very economical and they're very smart about how they do things and how they use energy. And yes, this is now that we're talking about it, it's an obvious way to do things, right? Is to utilize mechanical transduction where it's available to do some of these tricks, so you don't have to make a bunch of ATP or GTP or something,
which is precious.
So
which
yeah,
is precious.
Right. Absolutely. Absolutely.
I mean, nothing is for free.
Yes, nothing is for free, and yes, I think some of these mechanisms, and Paul says the same thing. You know, my partner Paul says the same thing to me, that gee, we may be onto some fundamental mechanisms here that just have been kind of overlooked. And I would tell researchers and students and anybody else entering this field, you have to keep your eyes open. You have to be willing to look at antisense, and you have to be willing to look at unpopular ideas. You know, the idea that Shine-Delgarno sequences in bacteria don't just happen at the beginning of genes, and they don't happen very often in general, but they happen in the middle of genes. They happen at the end of genes.
They do.
20% of bacterial genes are leaderless, have no leader at all, have no spacer, no untranslated region. It's just boom, 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 gene, 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 and they overlap usually by either one base or 4 bases usually. So they don't just butt up against each other, they actually overlap slightly.
Right, and then are cleaved later.
Well, there's a stop codon, but then to get to the start codon, you got to back up 1.
Oh, so it goes through, clips,
it goes through
a little bit, and then goes again.
And frame shifts. Yeah, you have to have a backwards frame shift of -1 to start up again. We think that's a deliberate mechanism to avoid read-through mistakes, because in bacteria, unlike higher organisms, which have multiple stop codons sometimes, bacteria don't use multiple stop codons. What they use is this mechanism that if you don't frame shift, and you just keep plowing right through there, you're going to start making nonsense protein very quickly. And that stuff's going to get marked for recycling right away. It's going to pop off the ribosome right away. way. So we think this is a mechanism that's pretty universal in bacteria, is to have this little frame shift go on from one gene to the next, so that if there's an accidental read through, it very quickly produces a nonsense protein of a type that can be easily, quickly recognized and dealt with. But yeah, I forget what I was going to say. I was saying that 20% of bacterial genes are leaderless, don't have a promoter, don't have a Shine-Dalgarno sequence. Actually, they do have a Shine-Dalgarno sequence in the preceding gene, at the end of the gene. The preceding gene is often purine rich right there at the end. That's your Shine-Dalgarno sequence. But people need to look at the fact that promoters don't just occur at the beginning of genes. They occur in the middle of genes, and they occur in the middle of genes on both strands. Sometimes on either strand, they can occur in antisense. We look at tuberculosis has more promoters in the middle of genes than at the beginning of genes. And if you look at where they occur on the reverse complement strand of protein genes more often than on the proper strand. So there's definitely something going on there. So my advice is, people, keep your eyes open, keep your mind open, and look for these things. Set aside whatever you learned in school, whatever the textbooks are saying, whatever the consensus viewpoint is. Set that aside for a minute and just use your own eyes, use your own mind, and come at these things fresh, and be willing to consider antisense, promoters that are out of place, Shine-Dalgarno sequences that are out of place. All these things are going to turn out to be important.
I couldn't agree with you more. I distinctly remember when I was learning molecular biology of looking at sequences coming out of yeast strains, and it doesn't have the right promoter, so that's a nothing. How many discoveries were thrown away with that mindset? How many? It's probably mind-boggling, and I'm sure I've done it plenty.
Absolutely.
But
absolutely,
I agree with you. I mean, since we're at a good spot for this question, and I just want to hear your answer. Being on the outside and then coming back, what was the thing that surprised you the most in a good way, and what was the thing that surprised you the most in a negative way?
Oh, that's a great question. I think the surprise in a good way is just the amazing amount of data that is being shared openly. So when I came back in 2014, started looking at what's out there, I just wanted to know what's out there. Well, I found all kinds of genomes are just publicly available now, and my mind was blown. So I think the amount of data that's being shared, not only genomes but RNA, high throughput proteomics data, and there's just these masses of data that are now showing up. That has been a pleasant surprise. It's not paywalled. It's not locked up in a lockbox somewhere. It's not, sequestered. So that's really wonderful. That's been pleasant to deal with. The unpleasantness. By the way, another pleasant thing is I think more papers have more and more authors now, which I think is a good thing. I think laboratory workers, technicians, postdocs, grad students are getting their names on some of these papers now. That didn't used to be a thing back in the day. You might get a footnote or something, back in the day. So that's been a good thing to see. And open access is a good thing to see. A disappointment, of course, is the opposite of open access, which is there's so much paywalled science still. That's kind of disappointing.
Most of it.
Yeah.
I mean, the reality of it is, it's a crime to the American citizen, a crime that they pay taxes.
That's
Those
right.
taxes fund the NIH and the NSF. The NIH and the NSF give the money to researchers at academic universities. Researchers use the money, make the studies, and publish them. They have to pay to publish them to a journal, that then the journal charges the taxpayer, if they want to read it, an exorbitant cost. And who does the journal pay? Well, they don't even pay the people that review the papers. So their overhead is fairly minimal. They have to have some on-staff editors and copywriters, but even now you have to do the copywriting yourself for most journals. You have to format it into the correct format. You have to do it.
Right.
So why do they need all this money? Number one. Number two, why are we even allowing it? And who gets screwed? The people that actually paid for the research in the first place.
Yes. I'm sorry, I had to move back inside to get electricity because my battery was running.
No worries.
Low.
I mean, we can get to wrapping this up, but I would love to speak to you in person. I'll
come to Tallahassee.
Yeah, well, that's easy enough. So let's wrap it up with this. What would you, because you've been through a very interesting road in science, what advice would you give to anybody of any age who is like, I'm thinking about science, I might want to do something in science? What directions would you recommend they go? What would be your Bazooka Joe advice for this?
Well, whatever other field and whatever other skills you learn some computer science, learn enough to write a little bit of code. I write in JavaScript. I mean, I can write in other languages, but I write JavaScript right in the browser. I do most of my work in Chrome. Yeah, and so my advice is get some bioinformatics skills. Bioinformatics is a golden era right now for bioinformatics. And there is a ton of low-hanging fruit for anyone who will go after it. So my top advice is, I don't want everybody in the world to learn how to program. I don't think you have to be a computer geek necessarily, but it's nice to have those skills so that you can do these kinds of experiments, because, in the bioinformatics world, unlike the laboratory benchtop world where you're lucky to get one publishable experiment per 6 months or per year, in the bioinformatics world, you can do a dozen experiments before your first cup of coffee. You can be 100 times more productive. So get some bioinformatics skills.
And I think that applies to anybody in any area of life sciences. Like if you're in biology, biochemistry, molecular, anything to do with the life sciences, you are at a point in the world where you need a little bit of bioinformatics just to exist, even as an experimental.
I agree.
And as for the coding, one of the first things I did after I stopped being a faculty member is I taught myself how to code. And
Sure.
It's one of those things that once you do it, you'll be so glad you did, because it is amazing how many things outside of science, just in your daily life, that a little bit of code can go a very, very long way.
That is really so true. And I am totally self-taught in computer science.
Me too.
And it is. It's something you can do. You just grab a book or a tutorial and just start. And it takes a while to get to that critical mass of where you can start to really do stuff and really start to write your own code. But there's, it's like a Karate Kid. There's a lot of wax on, wax off. And but just do it. Do the wax on, wax off.
How
Just
many
shut up and
times
do it.
can you write "Hello World"? I mean, this is.
Yeah, exactly. But you got to do it, right? You
But you
can't go
have to
around it. You
do it.
got to do it.
But you have to do it, and there are tons of free online courses on YouTube for any language you can think of. I mean,
That's right.
I got into C++ because when you're trying to do really large data sets and things like this,
Yeah,
C++
yeah.
is almost essential. But, and I just liked it because it's a language that's very structured. It's kind of a what my way or the highway language. I kind of like that. But I can understand why people like Java and like Ruby and like these Perl, even, which is now dead, Python, which is the new Perl. I understand why people like it because it's so flexible. It's so common language written. And once you learn 4, 5, 6 different modules of using it, you can actually be
Mhm.
pretty good right away.
Yeah, and I think you would agree with me that if anybody's listening to this and they're wondering what language should I learn, it doesn't matter really. They all do the same things, and you can do stuff in any language. So to just pick one. I like JavaScript because it's built into the browser. It's the language of the internet, and
It's the
I
language
can
of the
do
internet.
JavaScript. So that's the only reason I pick it. But I learned on C and C++ first. Those were my first languages, and I uh, went and looked at Java the minute it came out, and then rejected it, and then came back to it later. It's a fine language for certain things,
Well,
but
Java was
C and C++ are good too. I mean, they're fine languages and very, very powerful stuff. And if you want to write fast code, of course you write C++. That's what the JavaScript interpreter runs in.
right, exactly. But yeah, so a language runs on top of a language. But uh,
That's right.
the JavaScript though is very versatile, and it's gotten. I mean, even though it hasn't changed all that much, what changes have been made to it have really improved its usability, uh, that and ease of writing and ease of debugging. That's one of the major pluses of JavaScript. Debugging JavaScript compared to debugging even Python, quite frankly, is quite a bit easier. But if you compare it to C or C++ debugging, well, there's no comparison because it's so much easier to debug.
Uh,
Well, on
yeah, I love. That's what made Chrome a big hit. It was people forget that Firefox ruled the world before Chrome came along, and Chrome stole that audience from Firefox based on, in my opinion, their excellent developer environment and the developer tools that are built into Chrome. It's shocking how good the tools are if you want to write code right inside the developer environment of Chrome. So that's something for people to keep in mind. There's step-through debuggers and all kinds of neat stuff right built into the Chrome browser. It's just waiting there to be used. It's free, it's reliable, it's rock solid, and you can do almost anything you want to do in JavaScript. But that's also true of a lot of other languages. So it just depends, what you're trying to do. If you're going to do a lot of work with AI,
Python,
there's a lot of great Python stuff. Yeah, get good with Python because AI. There's so many libraries and tools that it almost doesn't make sense to use any other
unbelievable,
language.
exactly. Because I'm okay at Python, like I can get by. I don't. It's certainly not my favorite language, but man, is it powerful! Like you can do a lot with it,
Yeah,
and the packages that have
yeah,
pre-done things for you are. I
very
mean,
powerful.
there might be more Python plugins than there are apps in the App Store, and that's not an overstatement. There may be.
Yeah, yeah, yeah. It
Well,
is
on
very
that
handy
note,
stuff. So,
Well, on that note, we can wrap up for today. I really hope to be able to speak to you again in the near future. I really enjoyed our discussion today. Thank you so much for your time.
You're welcome.
All right.
Welcome. I enjoyed it.
Talk to you soon.
Okay. We'll talk to you soon, Randon.
All right. Bye-bye. Yeah.